of Almighty Allah. Good evening, all. I am Saida. It's an absolute pleasure to welcome you all to this incredible event hosted by the SCRMP chapter of Afghanistan. Today is not a special day as we gather to not only expand our knowledge but also celebrate the power of collaboration between brilliant minds from Afghanistan and India. Following that, we will have the main presentation by Mrs. Nagina Balali, focusing on the development of cryogel microcarriers for local drug delivery to glioblastoma. The Society of Clinical Research and Medical Professionals, SCRMP, a registered society based in Telangana, India. Ms. Nagina Balali is a former lecturer at the Faculty of Pharmacy, Kabul University, where she served in the Pharmaceutics department.
Assalamu alaikum and good afternoon to all of the students and ladies who are listening to my voice. And I hope everybody is doing well and you have opened your fast today. Also, today my topic is about development of cryogel microcarriers that are intended for local drug delivery in glioblastoma patients. Cytoma. Usually the patients who have glioblastoma multiforme, they have very poor prognosis and the overall survival for them is 18 months or less than 18 months. Picture of an astrocyte. It's usually a glial cell, and glial cells have different types. Look at the challenges that has been in the path of glioblastoma treatment. Here in this picture, if you see, there is a patient who had surgery and the tumor is removed, and here around the resection cavity where the tumor is removed, the concentration of glioma stem cells is very high. You might know whenever a surgery is done, the doctor will remove some extra tissue from the brain, but because brain is a very vital organ in our their active ingredient is carmustine, and they are locally delivered to the glioblastoma. After the patient has removed the tumor, these wafers are implanted inside the resection. Although we said before that there is a gap of 3 to 4 weeks, so we would like the drug to be released in a sustained manner, in a controlled manner for a long period of time period of 1 month or even more than that. So the cryogel microcarriers are actually matrices of gels that are formed by polymerization at temperature under 0 degrees centigrade. In this picture, you can see that they have a microporous structure. They have plenty of pores. SPA is 3-sulfopropyl acrylate. This will give a negative charge to our microcarriers, and because we wanted to load a high payload of brexpiprazole, so we increase the amount of SPA in our formulation. Freeze our cryogel microcarriers on higher temperature slowly. So when the ice crystal grows bigger, when they melt, so they will leave larger size of pore. From the cryogel microcarriers, first we studied with light microscopy, pH 3.7, as I said it. So, and as I mentioned before, that we had optimized. I'd like to add from the research that has been done before on brexpiprazole was this: that the clinical dose of brexpiprazole is 0.0167 to 0.667 milligram per kilogram. Add up that electrostatic interaction between positively charged brexpiprazole and negatively charged cryogel microcarriers. It is the key for the prolonged drug release because the negative charge on cryogel microcarriers. That's all from me, my supervisor, and the lab that we had worked, and this research was supported by Council for Academics.
Thank you all again for joining us today. We look forward to a fruitful and engaging discussion. Thank you so much.
I'd like to have a conclusion with Ms. Rabia John. I think she's present in here. She's our dedicated faculty advisor in SCRMP, and I would like to call her for final remarks and conclusion of today's webinar. Thank you so much for participating. It was a really great webinar and really enjoyed each and every topic that we have discussed tonight. Thank you everyone for participating and joining in today's session. Please fill the feedback form, and goodbye. Have a great night.